All right, so today I'm going to summarize for you some of my preliminary findings about the importance of phosphorus and arbuscular mycorrhizal fungal communities in performance of Gaillardia aristata across the northern tier. So first, arbuscular mycorrhizal fungi, or AM fungi, or AMF. They are symbiotic fungi that colonize plant roots. And this is a very widespread symbiosis. It's found in about 80% of land plants. And it's also believed that it allowed plants to colonize land 450 million years ago. So it's important for the evolution of plants as well. And the way the symbiosis works is that the AM fungi receive carbon from the plant in the form of lipids and sugars in exchange for nutrients or services. And here on the right, I have a picture of some typical AM fungal structures that you would see inside of a root. Okay. So phosphorus is an essential nutrient for plants, but it's also highly immobile in the soil, which often makes it limiting for plant growth. So that's one reason that AM fungi are so important for plants. They can extend out from the roots, you can see in this image here, and mine the soil for phosphorus that the plant is unable to access, either because it's outside of the root's phosphorus depletion zone or because they're getting into smaller soil pores than the root could. So AM fungi can provide up to 100% of a plant's phosphorus requirements, but plants can also acquire phosphorus directly from the roots. So there are cases where AM fungi might be less important. And I also want to point out that there is a high spatial variation in phosphorus. So you can see there's areas where soil phosphorus is higher and areas where it's lower, and this varies with geography. So the questions that I want to ask then, since we have good reason to believe that phosphorus should be important in structuring AMF communities, since it is one of the primary ways that those fungi benefit the plants and therefore receive rewards, how do AM plants and AM fungal communities differ with changes in soil phosphorus? And then also, how do AMF communities from high and low phosphorus soils have similar capacity to provide phosphorus to their hosts? Or are they influenced by the soil environment in which they were assembled? So to answer these questions, I'm studying AM fungal communities in the roots of Gaillardia aristata, also known as blanketflower. And this is a perennial wildflower native to the northern Great Plains. And it's also a species of special concern in Minnesota and probably elsewhere as well. And this is mostly due to habitat loss. So for many plant species, we don't really know the extent to which they actually benefit from mycorrhizas. And for blanketflower, there's conflicting evidence. There's anecdotal evidence that, that this plant displays high mycorrhizal responsiveness, which means that it grows much larger when colonized by AM fungi. And I have a picture here of what this would look like. So these two plants are the same species, and they both have access to the same amount of phosphorus in those pots, but the plant on the left has no mycorrhizas, and the plant on the right is mycorrhizal, and you can see how much of a size difference there is there. And that's due to the mycorrhizas that are providing more phosphorus to the plant than the plant could access on its own. So to answer those questions that I posed, my research examines AM fungal communities in the field and in the greenhouse. And so this image just kind of summarizes my research. So in the field, I can ask questions like, what do AM fungal communities at different sites look like? Are AMF communities at different sites different from each other? And then what is the extent of the relationship between different measures of fungal biomass and plant performance. And then if I take if I take the soil associated with those plants in the field, which as you can see, if we looked at them at much higher magnification, you would see that there's a whole community of different AM fungi there. If we took those communities and transplanted them to the greenhouse, what would they look like under different soil phosphorus conditions? And so for both the field and the greenhouse, I did analysis, various types of analysis to better understand these relationships. And I also want to point out that that each plant in the field had both plant and soil nutrient data collected. So then my hypotheses are, if mycorrhizas are important for blanketflower, then mycorrhizal plants will have greater biomass and phosphorus than non-mycorrhizal blanketflower. Also that if phosphorus is important for structuring AM fungal communities, that since sites that differ in phosphorus will also have different AM fungal communities, and phosphorus fertilization should shift AM fungal community composition. And then lastly, if AM fungal communities differ in their ability to provision phosphorus, then blanketflower inoculated with AM fungal communities from low phosphorus sites will have higher phosphorus content and higher Hyphal biomass in the soil. So going back to this map of the variation in soil phosphorus, the research that I did focused on this area here across Montana, North Dakota, and Minnesota. And you can see that towards the western end of that range, there's generally higher soil phosphorus levels and generally lower soil phosphorus levels to the eastern end. So then for my field study, I visited 12 sites in Montana, North Dakota, and Minnesota. And you can see on this map that each red star represents a site, and they're generally all at the same latitude, just on a longitudinal gradient. And this setup is interesting because there are very few studies of AMF communities across geographic distances looking at the plant species, so this is exciting. Most studies are just at a single site or just a few sites. We don't really get a sense of what kind of variation there is with geography and what might be driving those differences. So as you can see here, at each site I collected 10 individuals of blanketflower, and I also collected associated soil, both to analyze the soil and also to use that soil to inoculate plants in the greenhouse with those same AM fungal communities. Okay, so if we return to this graphic, I'm going to focus on the field portion of my research first. So after we collected all those field samples, we took them back to the lab and analyzed them. We measured hyphal length in the soil to understand how much fungal biomass there was. So basically we extract the hyphae, which are the fine thread-like filaments of the AM fungal body that spread through the soil. And so we extract them from the soil samples, we stain them, and then we look at them under the microscope to quantify their length per gram of soil. And then to understand what fungal species are at each site, we first extract the DNA from the blanketflower root samples, and I also extracted DNA from the field soil samples. And then we use PCR to amplify the ITS2 region, the internal transcribed spacer 2 region of nuclear ribosomal DNA, and that's it's part of the fungal genome that's highly conserved, but still variable enough that you can see differences between species. So after that, we use Illumina high throughput DNA sequencing, which uses barcoding. And in that technique, the barcode is a sequence of DNA attached to the sample that uniquely identifies each sample. So then samples are pooled and sequenced, but they're still distinguishable because each sample has its own barcode attached to it. And then after you do that meta barcoding and sequencing, then I used the Pipit's pipeline, which is a piece of bioinformatics software for fungal genomes. And I use that to identify OTUs, or operational taxonomic units, which are a proxy for species. Okay, so I'm going to share with you some of my results here. So first, soil phosphorus varied across the field study sites. And you can see that on the x-axis that the sites are organized from west to east, so showing that longitudinal gradient. And we wanted to have sites, you can see in general that there's higher phosphorus sites to the west and lower phosphorus sites to the east, which we were expecting. And we were trying to find lower phosphorus soils in the west and higher in the east as well, but we didn't really succeed in that. Everything out in North Dakota and Minnesota is really low. So we did not succeed there, but you can see there is a range of soil phosphorus levels. And then I also want to point out that in this graph, the color of the box plot indicates how we categorized the site. So we categorized them as low P sites or high P sites, and we use that for the greenhouse experiment that I'll talk about next. So in the field, we also found relationships between plant P, soil P, and plant biomass. So you can see that as soil phosphorus increases, so does plant P content. And then also as plant P content increases, so does the plant biomass. So this indicates that phosphorus could be limiting for blanketflower. And then we also have data showing that plant phosphorus increased with hyphal length in the soil. So that's a good indicator that that the hyphae are providing more phosphorus to the plant, since there's more biomass there for them to do that with. Okay, and then I used non-metric multidimensional scaling to visualize differences in AM fungal communities by site in the field. So in this ordination, each point is an AM fungal community associated with a single plant's roots. And here I'm showing 5 plants per site. So the closer the 2 points together are, the more similar their AM fungal communities are. And then also, you can see this legend here. I arranged the sites from top to bottom, west to east. And then triangles are symbolizing sites that are in Montana. Circles are symbolizing sites that were in North Dakota, and squares symbolize sites from Minnesota. And also I want to point out that symbols that are shaded indicate that those sites were categorized as high phosphorus sites, and symbols that are not shaded in were categorized as low phosphorus sites. So then there's a lot going on here, but you can see, for example, if you look at Bicentennial Prairie, which is this light blue color on the right-hand side of the graph, so those samples within that site are more similar to each other than they are to other sites, except that they overlap with the samples from, or the communities from B Ranch, which is not too surprising actually, since those sites are probably less than 5 kilometers from each other. And then you can see that generally the samples on the left are from Western Montana, and the samples on the right are from Eastern Montana, North Dakota, and Minnesota. And there's one site, Kleinschmidt, in the middle here. You can see it's the green triangles, and it's kind of a weird one because it's a site in Western Montana, but 2 of the samples from that site cluster with the other sites in Western Montana, but the other 3 are clustering with the sites in eastern Montana and North Dakota and Minnesota. So part of wrapping all of this up for my master's will be finding out why that is and why there's all these patterns in the communities that are causing these clustering patterns. Okay, and then overlaid on top of the ordination is a biplot showing some environmental variables that we collected as well. So these are the environmental variables that were correlated with the axes of the NMDS, and the length of each line or vector indicates the strength of the association. So therefore, longer lines are more strongly correlated with the differences among the AM fungal communities. So you can see that soil phosphorus and elevation are really strongly correlated with axis 1, as are magnesium and longitude. So we're seeing some, it looks like we're seeing some geographical structuring there of these communities, but there might also be an impact of soil phosphorus. And then one other thing I wanted to point out about this is that if you look, it's actually the shape of the clustering is actually kind of a backwards S shape. So it seems like that the second axis, axis 2, the vertical axis, is capturing differences within a geographic region, and then axis 1 is capturing differences across a geographic region. Okay. So to summarize the field results, we found that plant phosphorus increased with soil phosphorus, that there is greater plant biomass associated with greater plant P content, and that also that plants with higher P also had higher hyphal length in the soil. And then for the AM fungal communities that we just looked at, they differed by geography, elevation, and soil phosphorus, as well as by site. So for the field, I can conclude that differences in AM fungal communities may have consequences for plants, such as their phosphorus content and their biomass, and also that relationships between hyphal length in the soil and plant performance are important. For example, it does seem that blanketflower benefits from mycorrhizas due to the evidence from the hyphal length data. Also, site differences are important in these AM fungal communities, and environmental gradients like geography and soil phosphorus may be important for structuring AM fungal communities. Sorry about that. Okay, so now we're going to move into the greenhouse portion of the research. And so I'm going to talk to you a little bit about how the greenhouse experiment was set up. So the types of inoculum, and so those inocular are coming from each site, and you can see that they're arranged by mean phosphorus level. So the higher phosphorus sites are at the top, and each site is represented by 10 pots, 5 of which are assigned to the low phosphorus treatment, and 5 of them are assigned to the high phosphorus treatment. And at the bottom, you can see that there are also 10 non-mycorrhizal control pots. They received an autoclaved mock inoculum instead of a field soil inoculum containing a live AM fungal community. So those will serve as non-mycorrhizal controls that contrast with the rest of the pots. So this is kind of just reiterating that setup. We have 2 treatments. There's the origin of the field soil inoculum. So their inocula were coming from either a high or a low phosphorus site from the field. And then there's also the phosphorus level of the soil in the greenhouse pot. So half were fertilized with phosphorus and half were fertilized without phosphorus. Okay, now I'm going to move into talking about some of the methods that I used for analyzing these data. So I used the same methods that were used in the field, but I also looked at acid phosphatase activity. Phosphatases are enzymes that catalyze the hydrolysis of organic phosphorus to inorganic phosphorus, which is the form that's available to plants. And acid phosphatases are produced by plants, fungi, and bacteria. But in this greenhouse experiment, we controlled for the plant and bacterial sources, so we should be able to get at differences just from the AM fungal communities. And so to measure this, I performed enzyme extractions and then quantified using a spectrophotometer. All right, so looking at the data from the greenhouse, here's a non-metric multi-dimensional scaling of greenhouse and fungal communities by treatment. And remember that each point represents one AM fungal community. So the AM fungal community from each pot in the greenhouse, basically. So here we expected to see the communities shift due to fertilization with phosphorus, but we really didn't see that at all. The only differences here are between communities based on soil inoculum origin. So you can see that to the left-hand side of the ordination that there's mostly communities from low phosphorus sites, and then on the right side, there's mostly communities from high phosphorus sites. And then while this greenhouse experiment was going on, which was for about a year, I took rough estimates of plant biomass about every month or so. And I did that by measuring the length of the longest leaf. So that's a non-destructive method of estimating biomass. So initially you can see, well, I'll explain the graph a little bit better. So you can see the top section is plants that did not receive any phosphorus fertilization. And then the bottom section is plants that were fertilized with phosphorus. And then the blue dashed line is plants that were non-mycorrhizal, so they did not have any mycorrhizas. And then the green lines are from the different soil inocula. So originally there was a fertilization effect where the plants fertilized with phosphorus were larger on average than those that were fertilized without phosphorus. But over time that difference disappeared except for the non-mycorrhizal plants. Within the non-mycorrhizal group, the plants assigned to the treatment without phosphorus fertilization were smaller on average than those that received the phosphorus fertilization. And then mycorrhizal plants were similar across their phosphorus treatments. You can see that eventually the non-mycorrhizal plants in the fertilized treatments caught up in size to the mycorrhizal plants. However, the non-mycorrhizal plants that did not receive any additional phosphorus stayed pretty small the entire length of the experiment. Okay, so looking at greenhouse plant pea concentration, I found that pea fertilized plants have higher phosphorus concentration in their roots and shoots than plants fertilized without phosphorus. And then also plants with mycorrhizas have higher phosphorus concentration than non-mycorrhizal plants. And there was no effect of the soil inoculum origin. Looking at the final biomass, you can see that the non-mycorrhizal plants were significantly smaller than all the plants with mycorrhizas, and plants fertilized with phosphorus were significantly larger than plants fertilized without phosphorus, regardless of soil inoculum origin as well. The acid phosphatase data showed a trend where plants with AM fungal communities from low phosphorus sites had higher acid phosphatase activity, but it was not significantly different from the communities from high phosphorus sites. So to summarize these results in the greenhouse, we saw that AM fungal communities differed by soil inoculum origin, but not by fertilization. P fertilized plants had a higher P concentration, and plants with mycorrhizas had higher P concentration than non-mycorrhizal plants. In terms of biomass, non-mycorrhizal plants were significantly smaller than all plants with mycorrhizas. Plants fertilized with phosphorus were significantly larger than plants fertilized without phosphorus, regardless of soil inoculum origin. And when looking at the interaction, only non-mycorrhizal low phosphorus plants differed significantly from the rest. And then finally, for acid phosphatase, plants with AM fungal communities from low phosphorus sites had higher acid phosphatase activity. So what I concluded from the greenhouse was that from biomass and plant phosphorus, mycorrhizas are important for growth in blanketflower, and that mycorrhizal plants have higher phosphorus and are larger. However, phosphorus fertilization did not change AM fungal communities, and but P fertilized plants were bigger and had higher phosphorus concentration. And then lastly, soil inoculum origin did not affect phosphatase production, blanketflower phosphorus, or biomass. So my overall conclusions, looking back at my hypotheses, the hypothesis that that mycorrhizal plants would have greater biomass and greater P content than non-mycorrhizal blanketflower was supported. And then the hypothesis that sites would differ in phosphorus and that they would have different AM fungal communities, that was also supported. However, the hypothesis that phosphorus fertilization would shift AM fungal community composition was not supported. Additionally, we did not see that blanketflower inoculated with AM fungal communities from low phosphorus sites had higher phosphorus content. And I'm still looking into the Hyphal length in the soil data, so that's unclear at this point. And there was a trend for that higher phosphatase activity in AM fungal communities from low phosphorus sites, but not significant. And with that, I just want to thank everyone who helped make this research possible.